Hi everyone, I'm Karolyn Infanta D, research scholar from Electrophysiology Lab. I'm happy to present my PhD work titled Development and Evaluation of Drug-Loaded Nanotheranostics Hybrid with a Multi-Modal Imaging Against Pancreatic Cancer. This work was carried out under the supervision of Professor Dr. K. Maheswari ma'am, the Dean Shashi, SASTRA University. Let's begin with the overview of pancreas. Pancreas is a 6-inch hockey stick-shaped organ located retroperitoneally between the stomach and the spine. The major function of the pancreas is to help in the digestion, which is exocrine cells, and the endocrine cells, which help in the hormone regulation. When the cells in the pancreas develop changes in the DNA and grow out of control, pancreatic cancer develop, forming a tumor. The two broad classification of pancreatic cancer is the exocrine tumor, which accounts for 95% of all pancreatic cancers, and the neuroendocrine tumor, which accounts for 5%. Among all malignancy, the pancreatic cancer is the most lethal one. Globally, it is the 12th most common malignancy worldwide and 6th leading cancer cause of death. The 5-year survival rate is less than 2 to 9%. The WHO states that there will be the steep increase of 1.8-fold surge over the next 40 years. The rise in mortality rate gives rise to the question why. This is due to the fact of later diagnosis of the disease and poor therapeutic outcomes. In order to overcome these challenges, our research focus to develop and characterize a multifunctional nanotheranostics carrier encapsulating a dual model imaging hybrid and a chemotherapeutic drug for the treatment of pancreatic cancer. To achieve this objective, first we need to design a carrier and incorporate a drug and the imaging agent into the carrier and test the nanotheranostics hybrid in both in vitro and in vivo conditions. In order to achieve the objective, Nanotheranostics hybrid system was synthesized and characterized. The choice of the carrier was chitosan and the choice of the drug was gemcitabine, and the combination of imaging agent was MRI and CT. In order to achieve this, the gold-coated ionoxide nanoparticles were synthesized, and these hybrid molecules are encapsulated into the carrier along with the drug. The drug acts as a chemotherapeutic agent, and the gold acts as a MRI CT contrast, and iron acts as a MRI contrast. Since the iron exhibits a magnetic property, when the external magnetic field is applied, the heat is generated, which is harnessed for the cancer therapy. The characterization was carried out where the TEM electron micrograph shows the nanotheranostics hybrid is a spherical morphology. The drug release studies was carried out for the nanotheranostics hybrid, where the 100% of drug release in 15 hours in all the different conditions like physiological pH, acidic, alkaline, and in presence of protein. In order to study the efficacy of the nanotheranostics carrier in vitro condition, 3D spheroid models were developed as using a matrigel as a basement membrane. Three different conditions were optimized: overlay method, composite method, and encapsulation method. In all three methods, the spheroids were successfully formed. As they progress, the tumor spheroid grows in size, fuses with each other, and forming the largest spheroid. The progressive increase in spheroid formation was observed in all three conditions, and day 10, the live data assay was performed for all three conditions, and the encapsulation efficiency method shows relatively high spheroids, hence is taken for the further. The cell viability studies were performed for the 2D versus 3D cultures. The nanotheranostics carrier shows relatively high cell toxicity in both 2D and 3D condition, which impels the better intake of the drug by the cancer cells. The gene expression studies were performed for both 2D and 3D cultures. The pro-apoptotic markers BCL2 in both 2D and 3D for the nanotheranostics treated group was downregulated, and apoptotic marker like BID and P20 was upregulated in nanotheranostics treated group. The in vivo efficacy of the nanotheranostics carrier was evaluated in a xenograft model in a skid mice. The results show there is a 86% of reduction in tumor volume in nanotheranostics treated group compared with that of the positive control. And also in the imaging hybrid facilitates a better viewing of tumor in the site in both MRI and CT imaging. Hence, our result concludes the nanotheranostics system has a superior effect in addressing the pancreatic challenges, and also the nanotheranostics based system combination could be further explored for the treatment of pancreatic cancer. This is a list of publication from the current research work. I like to chance thank the Chancellor, Vice Chancellor for the infrastructure facilities and permission to carry out my research in the SASTRA Deemed University. I'd like to thank my research supervisor for all the guidance throughout my project, and I'd like to thank Dr. Ravi Kumar for the input in during the research. I'd like to thank the Nano Mission for the funding. I'd like to thank all my colleagues and their support for the successful completion. Thank you.